Hello everyone. Today we are going to discuss cell cycle. As you all know that the life of a cell or life of an organism or the entire biological system is dependent upon one very basic thing, that the one cell divides into two cells and it keeps on dividing, and then it forms more cells so that the life can propagate from one cell to the other. Or in case of a multicellular organism, the different types of cells are formed, and different cells that keep on dying, they are replaced with new cells. So in this way life can propagate. So this process, that one cell has to divide into two cells, it needs some kind of preparation, and after that preparation only that cell can divide into another cell. And this entire process we call as the cell cycle. Now, as you can see that the famous scientist Francois Jacob, you have heard his name, Jacob and Monod, I think during the discussion for the lac operon. So Jacob had a famous saying that he said that the dream of every cell is to become two cells. So what he wanted to mean was that the ultimate idea of a cell or ultimate goal of a cell is to divide into two cells. This is the basic primitive idea of a cell. This is not only the basic primitive idea of a cell, but this is the basic concept of life, to make a thing similar to itself, that is to reproduce. So cells also what a cell wants to do is to become two cells. Now, we will see that not all cells will always divide. Some cells will become specialized and they will remain as such, maybe indefinitely, maybe for some time, and may not divide after some time. But that basic nature or the basic tendency to divide, that will remain. That can be shut down for some time also. Anyway, so that we will discuss now in the discussion of cell cycle. So let us get an overall idea first, and then we will go to the details. As you can see, that cell cycle is a series of events. It is not a single thing, it is a series of events that take place in a cell, ultimately which will lead to the division of the cell. And this division of the cell will have two important parts. That one cell, if wants to go into two daughter cells, the most important material that it wants to propagate into the two daughter cells is its DNA. So in the cell cycle there will have to be something or some events that needs to divide or duplicate the DNA. That is, the process is known as the DNA replication, as has been already discussed. And after the DNA has replicated, the chromosomal or the genetic material has replicated into two sets. The other parts of the cell, that is the cytoplasm, the organelles, the important proteins, they will also be formed sufficiently. So as we can see in this diagram, that in different phases, you can see in this diagram that in different phases the nuclear or the genetic material is getting divided in the interphase. It is the preparatory phase, and once the entire sets of things including the genetic material and all other organelles and cytoplasm and proteins and all important things that are produced in sufficient amount, then the cell divides into two daughter cells by the process known as mitosis. We go into the different phases, as we have already discussed the different phases of the cell cycle. So in the eukaryotic cell cycle, we see there are two important phases, as I have said, that one is the mitosis or the M phase, where actually the division occurs, and then the interphase, where the preparation for the cell takes place. Other than the M phase or the mitosis, where actually the chromosomal or the genetic material is separated into the two daughter cells, the actual division of the cell membrane and the cytoplasm into the total two divided cells, that process is known as cytokinesis. So the mitosis and the cytokinesis, these together will divide the cell. So this part is the mitosis, and the cytokinesis forms the entire cell division. But the preparation for the cell division, that is the other parts of the cell cycle, that is the interphase, and which, as you can see, has 3 different phases, that is the G1, S and G2. Each of them will have some specific tasks, and sometimes some specific cells can go into, from G1 phase, can go into a special resting kind of phase which is known as G0, to which we will come later. But otherwise, in general, a cell cycle can be discussed as I have said, the interphase, which is G1, S and G2, and then the M phase or the mitosis, along with the cytokinesis, which occurs at the end part of the final part, along with the mitosis towards the end part of the mitosis, as we will see. So in this cycle we can see that the M part proportionately is much smaller, that is the cell division. The actual division occurs, or it takes a much less proportion of time of the entire cell cycle, and it ultimately leads to division into 2 daughter cells, and each new cell will again now enter into the next cell cycle of its own, where it will go through the G1, S and G2, which together constitutes the interphase or the I, as mentioned in this diagram. And sometimes some cells can go out of this cycle of preparation and division from the G1 phase in presence or absence of some specific signals. It will go to G0 phase, which is kind of a resting phase. Many differentiated mature cells, they remain in the G0 phase and do not keep on dividing further. So if in this table we can see some of the basic things that occur in all these individual phases. So in the mitosis, or M, the last thing, the mitosis, you see that the cell grows so much that now it has all the material, including genetic and organelle and other things, sufficiently so that it can now be supplied to 2 daughter cells. So at this phase the cell divides, and divides into 2 daughter cells, and the preparation is the interphase. In the G1 the cell increases in size. The G stands for gap, so G1 means gap 1. So in this the cell increases in size and different things are produced. Then the S phase, which is most important, which is the synthetic phase. Synthetic, so S. Here actually the DNA replication takes place. The most important thing, as I said, that the genetic material has to be divided into 2 groups. That occurs in the S phase. And then finally the last part of the interphase, that is the G2, the gap 2. Here the DNA that has been synthesized, that has been divided, it has been checked that there is no error, there is no DNA damage. You know how the DNA errors or the DNA damages are corrected by different repair mechanisms, etc. And it is finally ensured that all the important proteins, enzymes, organelles, they have been produced. They are ready for entering into the M phase. And between each of these phases, from G1 to S, from G2 to M, there are specific checkpoints, which will ensure, we'll come to that later, which will ensure that everything is okay, so we can now move on to the next phase. And G0, the cell leaves the cycle and stops dividing and remains in the resting phase like that. So you can just stop the video, and if you want to write it down, you can write these things down from this table. This is in a very short, in a nutshell, what occurs in each individual phases. So here again in this diagram we have just in a very jumbled up manner we have shown what exactly occurs in each individual stages. The mitosis or the M phase is discussed in more detail. Now here you can see that the M has been given more proportion, but actually, as from the previous slide, you know that the mitosis or the M phase actually takes a very little part. So if it's around 24 hours in an average, you can say, about to complete an eukaryotic cycle, the M phase takes only around 1 to 1.5 hours, not more than that. So the rest of it is the entire interphase, that is the G1, S and G2. Only around 1 hour of the 24-hour cycle, 20 to 24-hour cycle is taken by the M phase. But here that has been magnified, and the M phase has been discussed. What actually happens in the prophase, you know, these things I will not go into detail because I think this has been discussed in the physiology. cell cycle, cell division I think has been discussed in physiology, so we will not go into detail here. You know, I think that the cell division is divided into mostly 4 phases, that is prophase, metaphase, anaphase and telophase. Here it has been shown what actually occurs in the individual phases. So in prophase, as you know, that all the genetic material after completely dividing into 2 sets. So during the S phase, your DNA has been divided into 2 sets. So now your diploid genome of the cell has been divided into quadruploid. You have 4 sets of each chromosome. Normally in a mature cell, you should have 2 sets of each chromosome. So 23 into 2. But now after division, just before the cell division, you have 4 sets of 23 chromosomes. Now each of these chromosomes, the sister chromosomes after division, they have all been jumbled up like some coils that have been jumbled up together. So now during the prophase, they start to declutter, they start to separate out, and the individual chromatins that are very thin, they now start to condense up. So you can separately identify individual chromatins into, and they start becoming chromosomes. So in the metaphase, this process is completed, and each chromosomes with sister chromatids, that is the 2 DNA, same DNA that has been replicated during the S phase, they lie together. They are joined with each other by some specific proteins like cohesins. We will come to that later. Specific proteins near their centromere, and along with that another thing occurs, that is the microtubules start to form, that also form 2 poles, and the sister chromatids, they remain joined at the microtubule. During anaphase, what happens? The cohesing binding, they get resolved, and the sister chromatids each move towards the separate poles. And during the telophase, along with the cytokinesis, ultimately the 2 poles divide into 2 daughter cells, and the nuclear membrane is also formed. So this is in a nutshell about what happens in mitosis. You all know that. Next slide. So we will not go into much detail of the cell division, because I think you know it. So let us go into the other phases of the cell cycle, that is the interphase. But before that, let us talk a little bit about the G0 phase. That is not directly the part of the cell cycle, but it is a way where some of the cells, they go out of the cell cycle and remain in a resting phase. Resting or quiescent or senescent. So they do not keep dividing. They are in a non-dividing quiescent resting phase. So some of the very important example of it are the neurons. As you know, the cell division of the neurons occur in the very early infancy and during fetal life. After a certain amount of age, that is 2, 3 or maybe sometimes 4 years maximum, there is no further division of the neuronal cells throughout your life. So the neuronal cells, they remain in the quiescent stage, they are differentiated, and mature cells, they have some specific tasks and they keep on doing that task throughout their lifespan. Very rarely they come back from G0 phase to again enter into the cell cycle and divide. They will keep living in that form, and if their DNA gets damaged or there is some external stress, or if it has become too old, if that cell has become too old, ultimately what happens is that cell is self-destroyed and it enters into the process of apoptosis. Okay, so G0 is the resting phase where typically the differentiated mature cells that has been differentiated for a specific function, they remain. The best example is neurons, which in a very early infancy the division ends and all the mature neuronal cells that you have, they keep themselves in the G0 phase and keep on doing the specialized functions that they are supposed to do. In the interphase, now we know it is known as the preparatory phase, or sometimes it is also called as intermeiosis, because the intermitosis, because mitosis is the M phase, that is cell division, that is the ultimate aim or dream of a cell as said by Jacob. But the preparation or the preparation to that is the purpose of interphase. So as I already said that interphase has 3 parts, G1, S and G2, and typically it takes more than 90% of the time of the cell cycle. In the G1 phase, as we can see, the G1 phase, it keeps on the preparation ready for the DNA synthesis that will occur in the next phase, that is the S phase. During this phase, what happens? The biosynthetic activities of the cell take place. Different proteins and different enzymes that are needed for the DNA synthesis, and other things like say purines and pyrimidines that are needed for the DNA synthesis, and all the things that are collected and synthesized by the cell during this G1 phase. These collections and biosynthetic activities, they are very slowed down during the M phase, because in M phase the aim of the cell is to divide. The other activities are almost shut down or brought down to a very low level. So these things again pick up speed and these things start to occur. So also another thing very important that occurs in the G1 phase, that G1 phase has a very high amount of regulation, because here you will decide that whether the cell is ready for the DNA replication to take place or not, because once you replicate the DNA, that is an irreversible process. Once you have replicated the DNA, you cannot come back. So you have to ensure that you have everything ready. The cell is ready for the division finally during the M phase. Then only it should enter into the S phase where the DNA will replicate. So that is a very important regulation that occurs in the G1 phase. Also, it will determine whether the cell needs to divide or not. If it needs to differentiate and mature and go into the G0 phase. That also is decided into the G1 phase. In the S phase, as we have said, the main thing that occurs is the DNA replication. One DNA is divided into 2 DNA. So 23 into 2 diploid genome is divided into 23 into 4. Each DNA is divided into 2. That is, it becomes into the quadruploid genome, and once you have 4 sets of every DNA, you have to divide. Somehow you have to enter into the cell division. And other processes like protein synthesis, etc., now again come to a very low level. It was high in the primary preparation in the G1 phase, but once you enter into the S phase, here your aim is to DNA replication. So other processes again come down to a low level. In the G2 phase, here what happens, the DNA replication is completed. So now again different protein synthesis and other rapid cell growth takes place, different organelles produced. Now the cell starts to prepare to enter into the mitosis. During this phase itself, the microtubule formation that you know is needed for the mitosis, the microtubule formation that forms the mitotic spindle, these things start to form. And here it will try to see whether the DNA is damaged. If there is damage, then I will come to it later, a very important protein known as P53 that will start to trigger a process where the damaged DNA is either repaired, or if the repair cannot be done, the cell enters into the apoptosis or the cell is destroyed. Because if the DNA is damaged and you divide the cell, then this damaged or this mutated DNA will enter into the daughter cell, and this mutated or damaged DNA will propagate, and that may lead to cancer or many other diseases. Now, if all these things are checked to be correct, and all the preparation, the proteins and the organelles are produced in sufficient amount, then the G2 phase will cross the G2M checkpoint and will enter into the mitosis and cell will divide. Next, in the mitotic phase, as I have said, which is the division of the nucleus, is also known as karyokinesis. This will occur, and in different 4 phases, that is the prophase, metaphase, anaphase and telophase, the cell will divide. The cell division, again, you all know, we will not go into much detail. So these are the 4 stages as we know. So through this and the little bit of detail of exactly what happens, I have already told you, the cell will divide. Again, in this diagram, you can see that what happens in prophase, that these spindles is being formed and the nuclear envelope breaks up and the chromosomes chromatin start to condense into sister chromatids which are joined together at the centromere. Then in the metaphase they are all aligned in the at the equator of the cell, and the two spindles that are formed, microtubules that go into two different poles. And then during the anaphase, as I said, the cohesion between the sister chromatids is dissolved by certain processes, and then the sister chromatids move by the pull of these microtubules and these spindles into two different poles. And then in the telophase, again two different sets of daughter chromosome go into the two poles, where again the nuclear envelope formed, and alongside simultaneously the cytokinesis or the division of the cytoplasm and by the indentation of the cell membrane occurs, and finally the two daughter cells are formed, which again enter into the next interphase of the cell cycle. In this diagram, as we can see, that the different things that are occurring in the different stages. It's a very beautiful diagram. As you can see that in interphase everything is being formed. In prophase, as you can see, inside the nucleus, the blue area, the DNA which is dispersed, it is starting to condense in pre-metaphase, pro-metaphase, it has condensed somewhat, and also you can see the mitotic spindles are starting to form, the green things. Now in the metaphase, as you can see, next all the chromosomes are condensed and they are aligned in the equator of the cell, and the mitotic spindles formation has been complete. And then in anaphase, as you can see, they are pulling the two sister chromatids, two sets of sister chromatids in the two direction. And telophase, as you can see, the two sets of DNA, they are in the two different poles. A nuclear membrane is being formed, and cytokinesis by the indentation of the cell membrane you can also see in the telophase step. In prophase, as I have already discussed, what happens, so if you want to write down the details, you can write down the details from here. So the summary or the bottom line of prophase is the nucleolus disappears, the chromatin condenses, the separation of the centrosomes and formation of the mitotic spindles, as we have already discussed many times. Next slide. Again, in the metaphase, what happens, you can write it down from here, you can make a note accordingly if you want. The most important thing here, that they go into the chromosomes condense, they go into the equatorial plate, and still in the metaphase the sister chromatids are joined together by protein complexes known as cohesins. This is very, very important. They are still joined together, but the microtubules have formed, the spindle fibers are formed, and they are towards the two phases, the two poles. So the bottom line of metaphase is chromosomes line up along the metaphase plate or the imaginary plane along the equatorial plate of the cell. In anaphase, what happens, there are some proteins, some enzymes known as, for example, separase, which will cleave the cohesin ring which was joining the two sister chromatids. Now that the cohesin has been dissolved by this enzyme, the two sister chromatids which are joined by the spindle of the two opposite poles, they will now start to be pulled by the spindle fibers, and they will split and move towards the sister chromatids will move towards the two opposite sides of the cell. So here again the bottom line is the centromeric connection between the sister chromatids are broken down, that is dissolved, and the sister chromatids now start to move towards the two poles of the cell by the movement or the pull of the spindle, mitotic spindle. Next, in the telophase, what happens, that the sister chromatids have reached into the two poles, and they again start to, from their condensed form, they again start to open up and again form the chromatin form, and the nuclear envelope starts to form, and the cell cytokinesis also occurs along with. Next slide. So in telophase, the bottom line again is the new nuclear envelope forms around the sister chromatids, two sets that are going to the two opposite poles, and the chromosomes again unfold back to the chromatin form, nucleoli again reappear, and the cell elongates even further. And along with the telophase of the mitosis, the cytokinesis now starts to take place. It occurs somewhat together, and this cytokinesis will divide the cell into the two daughter cells. As you can see that the telophase and the cytokinesis, they are occurring more or less together, that the nuclear membrane reform and the nucleoli form, etc., etc., and the cell is cleaved into two by pull or by the action of again some different filament types of molecules known as myosin 2 and actin. They will help in the cell membrane to cleave into indentation and then cleaving it into two daughter cells. In this diagram again, the little bit of details and the little bit of summary of what exactly occurs in each different phases of the cell division is been given. So you can again use it for making some short notes, or you can just go through it to have a just summary of what is happening of all the things that we have already discussed. So now after we have seen that what exactly occurs in each phases of cell cycle, that is the phases of interphases, that is the G1, S, G2, and during the different parts of the mitotic for the M phase, that is all the four phases. Now we have seen what happens in the cell cycle. Next we need to move on to the very, very important topic of how this cell growth and cell cycle is controlled. What is the regulation? This, as you can understand, that this has to be regulated very, very tightly, because any error in any one part will lead to a catastrophe in the life of this, not only the cell but of the entire organism. So entry of the new cells in the tissue population is largely determined by their proliferation rate, and cells will leave the population by cell death or differentiation. So in a tissue you have a set of cells. So new cells are entering into that population through cell division of the cell cycle, and from that population some cells are going out because they are being differentiated into the specified cell, and they will now go into G0 phase. Or if it has some defect or it is become too old, then the cell will die and go into cell death. So this entire thing is occurring. New cells are coming into the pool. There is a pool of the cells in that tissue, and cells are going out because they are getting mature or they are dying. So these things, that the proliferation of cells in a particular tissue, how many new cells are being formed, they are very, very important, because continuously there may be an injury, there may be cell death that has to be replenished by new cells. But you also do not want to replenish too many cells so that there is an unchecked entry of more cells. So this has to be regulated by many, many regulatory factors and many, many regulatory mechanisms. So there are many important biochemical mediators, different chemical factors, environmental factors, and mechanical factors that some of will help or push the cell towards more growth or more division, or some will oppose it and take it out. Some will help it to differentiate into specified cell, and some will help into going out completely through the cell death or apoptosis. Next slide. So this thing you can see that some of the cells, they are going out, proliferating, more and more cells are produced, and again from that population some cells are differentiating into specified cells. They now enter into G0, they no longer divide. Some cells, they may be too old or damaged, then they will go out of the population into cell death or apoptosis. And continuously from the stem cells, which you know, you have heard of it, the stem cells which can divide into new cells, the stem cells, they are dividing into produce new cells of the cell population. So it has three different ways to go. One, it can further divide and proliferate. Some of the cells will divide and proliferate, enter into keep on going into through the cell cycle and cell division. Some will differentiate and stop dividing, enter into the G0, and some will die through apoptosis. So the proliferation and cell division and continuation of cell cycle is different in different types of cells. Some will keep on divide probably throughout your life, and some will stop dividing at very early stage of your life. Next slide. So depending on those characters, the cells can be divided into 2-3 different types of cells. For example, one group is known as labile cells. They are continuously lost. The cells, some of the cells die and some new cells are continuously being formed, and this cell cycle of these types of cells, they keep on continuing almost throughout your life. For example, you know the skin cells, the epidermis cells. The skin cells keep on dying, we shed skin cells and new skin cells keep on coming from below. The GI tract, the epithelial cells, the intestinal epithelial cells, they, as you all know, keep on dying and new cells are continuously being formed. Blood cells from the bone marrow precursor cells, they continuously, as you know, the lifespan of a mature RBC is 120 days. So you need to, every 120 days, every day, many of the blood cells that has been formed 120 days ago, they die, and you keep on need to replenishing by producing more and more RBC cells. So these are some of the cells that keep on dividing. Then there are some cells which are known as stable cells. They remain mostly quiescent in the G0 phase and very little replicative capacity. They usually do not divide in normal condition, but they still remain some capacity to divide. These are like the liver cells or the hepatocytes. Usually in a normal person, they will not divide. But you need to remember that these cells have a very good capacity to divide if needed. So therefore, these are known as stable cells. They do not divide normally. But then there are a third group of cells which are known as permanent cells, which once differentiated into the mature and a particular functioning cells, they almost do not divide and they almost lose all capacity to divide even when needed. These are examples of these cells are neurons and cardiac myocytes. So one thing you can understand from this, that if you lose neurons or your cardiac myocytes, say by an acute myocardial infarction or by a stroke, it is very difficult to replenish these cells because you only have mature cells and these cells cannot divide again. They have almost lost their capacity to divide. On the other hand, your hepatocyte cells, liver cells, and so liver cells, you know, if you lose a part of the liver, you can still regenerate it, and this is how the liver transplant acts. Because someone has his or her liver completely destroyed, completely lost, you just need to take a little piece of liver from a normal healthy donor who is matched, the immunologically matched normal donor, and that little piece of liver tissue can again divide into forming a sufficient amount of liver tissue for the person who has accepted the liver transplant. So that capacity is still present, but in a normal condition the liver cell will not divide. So both stable cells and permanent cells are remaining in the G0 phase, but the permanent cells will almost bound at the G0 phase, and even if it needs, it cannot come out of the G0 phase usually. Next slide. So here again we see what are the different parts of the cell division. We have already discussed this. Next slide. So now we go into the normal cell proliferation, how that occurs. Next slide. The regulation mainly of the cell cycle, as we can see, now depends on how the preparation is complete and how individual parts can cross one phase of the cell cycle to the next phase when everything is completed. And as we have discussed this term, that is the checkpoints, that from G1 to S, from G2 to M, etc. These are some important checkpoints. So now these checkpoints and these individual phases, which phase is now the cell cycle is and what has to happen in that particular phase, etc. And now it has converted from which cell cycle phase to which cell cycle phase. These things depend on two particular group of molecules known as cyclins and cyclin dependent kinases, CDK. The CDKs are some enzymes. Kinase, as you know, they phosphorylate some important proteins, and by phosphorylating some important proteins, they regulate the cell cycle and they regulate the checkpoint. And cyclins are proteins which in turn activate specific cyclin dependent kinases. Now cyclin dependent kinases or CDK, they are always present. They are constitutively present in the cell. But cyclins are proteins that during different phases they are synthesized and rapidly again when their function is gone they are rapidly degraded. So when a particular cyclin is formed and present, they will activate a specific cyclin dependent kinase. The kinase then will phosphorylate specific proteins and specific things will happen. And then next after some time the cyclins will be degraded and the CDKs will again be inhibited because the cyclins are not present. And then again that function will stop and the next set of cyclin will come and next set of CDK will be activated and next set of thing will start to happen. Next slide. So now we will go into the regulation of the cell cycle, eukaryotic cell cycle through the cyclins and CDK mainly. Such regulation of the cell cycle, they involve many processes and you can understand that this is very crucial for the survival of the cell and so that the normal kind of cell is propagated. And this involves many things like detection and repair of genetic damage as well as prevention of uncontrolled cell division. This is very important because and each different phase, if you once move from one phase to another, this is almost irreversible. One thing, once you go from G1 to S, it becomes almost irreversible. You cannot go back to the G phase. Once you enter into S phase, you have to divide the cell, have to divide the DNA or replicate the DNA. So this has to be every checkpoint, every process of moving from one phase to the next has to be regulated very tightly. So as I have already told you that two different groups of proteins, that is the cyclins and CDKs, they are very important in this overall regulation. The cyclins are regulatory subunits that will regulate the action of the enzyme, that is the CDK, that is the cyclin dependent kinase. The CDK are the catalytic part. Now the CDK usually remains inactive in absence of the regulatory part, that is the cyclin. Only when this cyclin comes and binds with the CDK, then only this CDK will be activated, and because it is a kinase, it will phosphorylate certain target proteins, some of which will be activated, some of which will be inactivated, and overall result will be progression of the cell cycle in a specific direction. Next slide. So here you can see that in the G1 phase there are certain CDK and certain cyclins. We will come into their detail and which cyclin acts where, etc. And when the G1 phase ends and it enters into the S phase, the cyclins needed for the G1 phase will be degraded. So those CDKs will again be inactivated. So the next kind of cyclin, that is for the S phase, they will enter, and next kind of CDK will be activated. And then the next phase, when we move on to the S kind of cyclin, will be destroyed. Again remember that the CDK is always present, CDK is not destroyed. Cyclins are produced and destroyed depending on the phase of the cell cycle. And when a particular cyclin is present, it will activate a specific CDK. So CDK is present always, but its action is getting activated and again inactivated depending on presence or absence of the cyclins. Now there are certain inhibitors also which can regulate these processes. There are two family of proteins or genes that can inhibit these CDKs, etc. One is known as CIP-KIP group, that is CDK interacting protein or kinase inhibitory protein, this family, and a second is INK4A ARF family, that is inhibitor of kinase 4 or alternative reading frame family. And what are the examples? The CIP-KIP, it contains some very important genes and then proteins, that is P21, P27 and P57. And P21 itself is activated by another very important protein which is known as P53. You all must know the name of these proteins. P27 is activated by another very important protein known as TGF-beta, that is transforming growth factor beta. So all these proteins, they inhibit different CDK and therefore they inhibit the CDK to act in such a manner so the cell cycle progresses. So essentially all these inhibitors, what they do, the final action is they inhibit the cell cycle progression. And by inhibiting the cell cycle progression, they all decrease or inhibit the chance of formation of tumor. Because if there is too much of cell division, too much of cell cycle progression, then it can lead to cancer. So all these types of genes, they are known as tumor suppressor genes. So both P21, P53, all these are examples of tumor suppressor genes. And on the second family that I've said, that the INK4A-ARF family, it includes some proteins like the P16, INK4A, etc. They also inhibit certain CDK proteins, etc. Here we can see that what are the specific cyclins and CDKs that act in specific phases and specific checkpoints. As you can see, cyclin D, it is the very important, cyclin D is the most important cyclin of the G1 phase. In G1 phase, cyclin D activates two different CDKs, that is CDK4 and CDK6, and they help in the activation or inactivation of certain proteins that help in the things that occur in the G1 phase, that is like the preparation, formation, synthesis of different proteins and preparation for the DNA replication. Then when that is completed and you cross the checkpoint, that is the G1S checkpoint, once your G1 phase is complete, you cross the G1 to S, that is known as G1S checkpoint. And a very important protein that regulates that checkpoint is known as the RB gene. RB gene. We will discuss a little bit about it. RB, or the retinoblastoma gene. It produces a protein known as RB protein, which is very important in regulating the G1S checkpoint. Once that checkpoint is crossed, the cyclin D level will decrease, cyclin will break down. So CDK4 and CDK6 will now be inactivated. Now the next phase, that is the S phase, will start, and here the cyclin E will start to act. Cyclin E will help you to cross that checkpoint and enter into the S phase. Cyclin E is very important, and CDK2 will act here. Also cyclin A has some role. And then from the S phase to G2, you can see cyclin A activating CDK1 will enter into the G2, and during the crossing of G2 to M, there is another checkpoint, that is G2M checkpoint. These are the two main checkpoints, G1S and G2M. Here at this checkpoint, you see the role of cyclin B with CDK1. And there are certain inhibitors that you can also see, which is inhibiting certain proteins. That is like cyclin D, CDK4 in the G1 phase is inhibited by the P16, P15, P18, P19. Of the synthetic part, that is the S phase, where DNA replication occurs, you can see inhibitors like P21, P27 and P57. As you can see, they inhibit the cell division. So they are inhibiting the CDK2 there. So next we come to the checkpoints specifically. As we have seen, there are two main checkpoints, but there are other checkpoints also. The two major checkpoints are the G1 to S, because once you enter into the S phase, you divide the DNA, and that is irreversible. And then G2 to M, where you divide the cell altogether. So before the DNA division, DNA replication, there is one very important checkpoint, and then before the cell division, there is another very important checkpoint. So cell cycle checkpoints, they consist of a network of regulatory proteins. As we have seen, they will ensure that the cell cycle progression at specific point is allowed, because they verify that what has to happen has happened or not. These are things they will decide that and then allow. We will come to them. Next slide. So as we can see, the two major checkpoints are the G1S, where from G1 to S entry occurs, and then the G2 checkpoint, that is G2M checkpoint, where the cell enters into the replication. And during the metaphase, or the metaphase of the cell division, there is another checkpoint which checks for the chromosome spindle attachment or not. But otherwise, other than that cell division checkpoint, the overall cell cycle checkpoints are mainly two important checkpoints, that is G1S and G2 to M. Next slide. So let us discuss the checkpoints. The G1S, this checkpoint is the most important checkpoint, and if you can say, it is like the rate limiting step. Like in the metabolic pathways, we have seen the rate limiting step. Here, if we can think of one particular step to be rate limiting, then it is this checkpoint, that is G1S. This is also known as restriction point, because as you can understand, again, once the DNA replicates, it is very, very difficult to stop the cell from dividing. So what we check at this checkpoint, whether the DNA is okay, enough resource for the cell division is or the cell replication is sufficiently present, all the enzymes, all the purines, pyrimidines and the sugars etc. what you need for the replication, is there sufficient? Has the protein sufficiently been produced? Is the environment for DNA replication sufficiently good? So if all that is okay, if there is no damage in the DNA, then this checkpoint will be opened. So now the cell will be allowed to move into the next phase. Next is the G2M checkpoint. So after the cell division, the DNA has replicated and all that has been done, now cell has to enter into the cell division. Here, before dividing the cell, you have to ensure that certain things are in order. This is G2M checkpoint. Has the DNA correctly replicated? Is there some mutation? Is there some damage in the DNA? If yes, then that checkpoint will be stopped, cell will not be allowed, it will be given more time so that the DNA damage can be corrected before it enters into the cell division. Because once the cell is divided, if it contains a mutation or damage, then from all the next generation of the daughter cells, that damage will be propagated and a damaged set of daughter cells will be formed, and which very well may lead to a disease or even cancer. Next, is the DNA replication complete? And is there sufficient proteins and other organelles and everything built up in sufficient manner? If all these things are, the answer is yes, then only this checkpoint will be allowed and cell will finally enter into the mitosis. There is also a specific checkpoint during the metaphase of the mitosis where the proper attachment of the spindle etc. is seen. But again, most importantly, the cell cycle checkpoints are, at least you need to remember these two, that is the G1S checkpoint and the G2M checkpoint. Next slide. So if we fail to monitor the processes, or if these checkpoints does not act well, then what will happen? The damaged DNA, the damaged cell will be, the defect will be propagated to the daughter cell, and that defect will be keep on propagating into all the cells that are now produced from the division of that daughter cell, and which will lead to many different diseases or even cancer. So what happens if these growth inhibitory signals that helps in the checking of the cell division or the cell cycle progression, if they do not act? Next slide. So there are certain genes, as we have said, which are known as tumor suppressor genes, which help in the inhibition of cellular progression or the cell cycle. They are very important regulators. I think oncogene, tumor suppressor genes, etc. has been discussed in some previous classes. These tumor suppressor genes, there are many types of tumor suppressor genes, but these tumor suppressor genes, they specifically regulate different checkpoints and different cell cycle stages, so that they ensure that the cell does not divide too much, and also cell does not divide when the cell is not ready or correctly positioned for the cell division. 4 important groups you need to know the names of, that is, one is RB gene or the retinoblastoma gene, which we have already mentioned. It is called the governor of the cell cycle. Whether the cell cycle progresses into the mitosis, that is governed by the RB gene. Then there is P53 gene, which is known as the guardian of the genome, because P53 helps to correct any DNA damage. So it maintains the integrity of the genome. These two are very important. Then there is TGF beta, transforming growth factor beta, and there is another group of contact inhibition, where one cell to cell contact, certain outside signal will tell the cell to stop dividing, etc. There are some proteins known as NF2 and APC. I will not go into detail of them. Next slide. So we will just discuss a little bit about the two most important, that is the RB and P53. Again, I think this has been discussed. I will not take too much time. So you can see what is written here, but most important thing is you can see that RB usually remains in a dephosphorylated or hypophosphorylated state, and then it binds to a transcription factor known as E2F, and this E2F transcription factor, they help in the inhibition of the cyclin E, so that essentially what happens, RB gene when dephosphorylated and active, it inhibits the cyclin E to be formed, And you can remember we have seen that cyclin E is the major cyclin for the S phase. So if cyclin E builds up, is formed, then S phase or the DNA division will occur. So if there are certain growth signals that are present, then those growth signals, if are present, which tells the cell to divide, they only will phosphorylate the RB protein, and on phosphorylate the RB will release the E2F, and then cyclin E will be produced, and when cyclin E is produced, the cell starts to divide because the cell's DNA division or the DNA replication takes place. And if this process is damaged, if the RB gene is damaged, then cell will keep on dividing, the DNA will keep on replicating without any check, without any regulation. And that may lead to many different types of cancers. And in fact, it has been seen that many cancers where the G1 checkpoint, that is the G1 to S, whether the cell can enter into the checkpoint of G1S, the synthesis of DNA, whether allowed or not. Many cancers have this checkpoint damaged, and in many of them, a large part of them, the RB gene is found to be damaged. As you can see in this diagram, see, here is the RB gene. The RB, when unphosphorylated or less phosphorylated, hypophosphorylated, binds to the E2F, as you can find in the diagram, E2F which is bound to the DNA. And when it is bound to the E2F, it inhibits the production of cyclin E or other cyclins of the S phase. But when there are specific growth signals, like you can see in this diagram, growth factors like EGF, PDGF, etc., if they are present, they will lead to phosphorylation of RB. This phosphorylation takes place again by certain cyclins and cyclin dependent kinases like cyclin D, CDK4/6, cyclin E and CDK2. So they instead will phosphorylate, and if it is phosphorylated, then it will leave the E2F, and then the cyclin E will be produced and the cell will enter into the S phase from G1 phase. And there are some growth inhibitors like, as we have told, TGF-beta P53, which inhibits the process of RB phosphorylation and helps the RB to remain active. So there are signals from both sides, activating signals, inhibiting signals, like any biological process, if you can see. So depending on at what particular point, what particular signal is predominant, the process will move or not move in that direction. If the growth factor signal is predominant, then the RB will be phosphorylated and inhibited, and cell cycle will progress. And if the growth inhibitor signals are predominant, then the RB will remain active and cell cycle will not progress. And finally, the P53 gene, that is also known as the guardian of the genome. This P53 gene, it has a very good capacity to, or the protein that is produced by the P53, it gets activated on certain conditions where the DNA is damaged or can be damaged. They are activated by anoxia, when there is oxygen is less, when there is certain oncogene signaling, and when there is directly DNA damage. So P53 protein can sense all these things, and whenever it senses all these things, it activates many other proteins, like it leads to activation of P21, it prevents the phosphorylation of RB, so it does all these things, and essentially it prevents the cell cycle checkpoints, and then it arrests the cell cycle and it gives cell to more time for the repair of the damage. So it gives time to DNA repair. Only when everything is okay, it will again allow the cell cycle to progress, and if the things are not, or the problems are not repaired, P53 will send the cell to apoptosis. So this is how the P53 protein helps in the cell cycle or the cell DNA genetic material to remain always in the correct form. Whenever there is a defect or damage in the DNA, it will not allow that defect to be propagated to the next generation of cells. So that is why it is known as the guardian of the genome. So when the P53 is normal, whenever DNA damaged, there is cell cycle abnormality, hypoxia, it will arrest the cell cycle, as you see. It will give more time to repair and then allow the cell cycle to restart. And if that is not possible, not correctly done, it will send the cell to apoptosis. So if in a particular individual the P53 is mutated, then what will happen? The DNA damage or any condition that can cause DNA damage or abnormality will not be stopped. The cell cycle will continue and the abnormal genetic material will go to the next generation of cell, which may often lead to cancer. So mutated P53 leads to a large variety of cancers, and in many cancers you can find there is some amount of damage in the P53 gene. So with this, I think we can complete our understanding of the P53. You can just stop the progression of the class at any point of the video, any point, and take a little bit of note that has been written. Thank you.